Okay, the second part of our Unit 7 Keystone review has us on number 5 of the review, and it says, use the reading to describe and give examples of different types of mutations. This is right in the reading packet. I want you guys to make sure that you're taking a look at that, but I'm going to quickly go through it with you. Mutations can occur randomly during DNA replication or due to environmental factors such as exposure to toxic chemicals. These would be called mutagens. If the mutation occurs in a sex cell, it can be passed on to offspring. So just mutations that would occur in like a skin cell wouldn't be passed on to your offspring, only if it occurs in your gonads, your testicles, or your ovaries. Mutations can be harmful, beneficial, or inert. And inert means that they could have like no effect on the organism. Types of mutations. Your point mutation is where a single base pair in a strand of DNA is replaced with a different nucleotide. The remainder of the protein sequence remains remains unchanged. So for example, AUG, ACU, AGA, these translate to MET, THR, ARG. That's the normal sequence. The amino acid the nucleotide that's changed here is the C, gets replaced with an A by accident. That's your mutation. This is going to result in MET, ASN, ARG. The rest of this sequence is going to stay the same. Nothing's going to change with the rest of the sequence. The change is only occurring at this single point, so that's why it's called a point mutation. Next up, we have a silent mutation. These are more likely to occur if the mutation occurs at the end of the codon. Okay, here ACU is what it's supposed to read. Instead it reads ACC. This results in the same amino acid being transcribed for. So you end up with the same amino acid sequence. There's no difference in the phenotype. A nonsense mutation codes for a stop codon. That's going to break the amino acid chain and make the chain shorter than it's supposed to be. Okay, so that's obviously going to be a more detrimental mutation than maybe just a point mutation, depending on what happens. And then a frame shift mutation causes the entire sequence to be shifted. This was easier to describe with a sentence. If I said the fat cat sat, if I take out the if I take out the T and shift the whole sentence upward, you can't even tell what that was talking about. Hef, at. Okay, so the whole sequence gets screwed up. Whereas if we just took out like the letter F, but kept everything the same, okay, you would still know it's something about a cat sitting. But if we take out the one letter and shift everything, that ruins the whole sequence. So those are your 4 basic types of mutations. Number 6, again, this is just a lot of reading. I want you to go back to the reading packet and use the reading to describe the current trends in biotechnology. Things that you just want to give yourself a little refresher on are genetic engineering, gene therapy, genetic counseling, gene splicing, transgenics, and recombinant DNA. This is any time you manipulate the DNA of an organism, and you change the DNA of an organism to get a desired result in the phenotype. That's biotechnology. That's any of these things on this list. All right, genomics is the science behind the Human Genome Project. The human genome was sequenced back in, I want to say 2003, but I could be wrong. I double check that with the reading. But they sequenced the human genome and they manipulate the genes for practical purposes. They're working on using this information for molecular medicine, DNA in forensics, and agriculture. You may have heard of genetically modified organisms. Genetic engineering is being used right now to try to produce medicines, cure genetic disorders, and modify crops. Excuse me. And then gene therapy is when specific gene sequences are inserted into a cell to replace a defective or mutant allele. So they're trying to cure diseases using gene therapy. Some examples from the Keystone Review. In order to study genetic mutation, scientists must study genetic material. Which statement best describes the genetic material scientists are most likely studying? So looking through all of the choices, they study alleles that contain chromosomes, which are RNA. That doesn't make sense. They study alleles that contain genes, which are chromosomes. They study chromosomes that contain DNA segments, which are RNA. Or they study chromosomes that contain genes, which are DNA segments. To help me answer this, I just kind of drew my little picture. I've got my DNA, I wind it up on little proteins called histones, get it all nice and organized, and turn it into a chromosome. So chromosome contains genes, which are segments of DNA. So D would be the most correct answer here. An inversion occurs along part of a chromosome. How does the inversion most likely affect the region in which it occurred? So to invert something means just to flip it over. So I gave myself in a little example sequence here, and I flipped part of it. All right, so what's going to happen? Recessive alleles become dominant alleles. That's not what happened. The information reads differently during transcription. That would happen. The coding sequence disappears. That's not what happened. Or frame shift errors are triggered after translation. Still the same amount of nucleotides, so frame shift errors wouldn't make any sense either. So by inverting my little sequence here, all I did was read the information differently. So B would be the most correct answer there. Okay. I feel like this is going to be an easy one for you guys because we speak often about the central dogma, but which label correctly completes the diagram? And if we look down to letter C, Y is transcription, Z is translation. What makes me sad though is that only 40% of students in Pennsylvania got this correct last year, and I think you guys are going to all get it correct. So there's that. All right, next up is going to be Unit 8. So good luck with Unit 7. Let me know if you have any confusion about anything, and we'll talk later.